Continuation of the enzyme chapter. We were talking about the non-competitive inhibition. So, binding outside the active site induces conformational changes in the enzyme molecule. This in turn affects the structure of the catalytic site and prevents its action with substrate. The rate of non-competitive inhibition is directly proportional to the concentration of the inhibitor and its affinity for the enzyme. With the catalytic site, binding outside the active site induces conformational changes in the enzyme molecule. This in turn affects the structure of the catalytic site and prevents its action with substrate. The rate of non-competitive inhibition is directly proportional to the concentration of the inhibitor and its affinity for the enzyme. So, non-competitive inhibition is not fully reversible. So, an excess of the substrate is of no use in removing the inhibition. Delocking of non-competitive inhibition can be accomplished by inhibitor binding substances called reactivators. They inactivate the inhibitor to pull it out from the enzyme. In non-competitive inhibition, Km will not be affected by the presence of the inhibitor, but V will be lowered. This type of inhibition may sometimes interfere with the dissociation of the ES complex. Uncompetitive inhibition. This is the inhibition imposed by an inhibitor which has high affinity for enzyme substrate complex only. So it combines only with the enzyme substrate complex and not with free enzyme. The uncompetitive inhibitor first facilitates tight enzyme substrate binding, then it binds with the substrate part of its complex and inhibits enzyme action. This uncompetitive inhibition mainly depends upon the affinity of the inhibitor for the substrate. It is also apparent that the enzyme substrate complexing is more effective in the presence of the inhibitor. Uncompetitive inhibition cannot be overcome by increasing the substrate concentration. The inhibition of placental alkaline phosphatase by phenylalanine is a typical example of uncompetitive inhibition. Next is the substrate linked inhibition. Substrate linked inhibition. Substrate inhibition is the inhibition of enzyme action by an excessive substrate concentration. Substrate inhibition is the inhibition of enzyme action by an excessive substrate concentration. Substrate concentration is increased here. Substrate inhibition is the inhibition of enzyme action by an excessive substrate concentration disproportionate to enzyme concentration. In this case, the substrate binds with the enzyme not only at the active site but also everywhere. So, it will not be subjected to catalytic conversion. Moreover, such an enzyme substrate complex makes the enzyme molecule inactive. Substrate linked inhibition disappears when substrate concentration falls to normal. So, what is happening here? The enzyme is there, but the substrate is binding not only in the active site but on the other sides also. So, it will eventually turn off the enzyme. Substrate inhibition is peculiar in that a substrate is able to inhibit enzyme action on its own molecules. This is less common. Next is the product inhibition. In some metabolic pathways, the product of a reaction may not be much different from the substrate of a previous reaction, which is one or more step behind. So, such a product may compete with the substrate for a previous reaction for the active site of the enzyme of this reaction. This is called product inhibition. This is called product inhibition, feedback inhibition, or negative feedback. Product inhibition, feedback inhibition, or negative feedback. Next is the end product feedback inhibition. The most important type of enzyme inhibition is the end product inhibition. It operates as a natural regulatory mechanism in cells. Some metabolic pathways have built-in mechanisms for self-regulation. In them, the enzymes of the initial reaction acts as a regular enzyme or pacemaker enzyme, and the end product of the reaction chain acts as an inhibitor of this enzyme. The regulator enzyme initiates. It operates as a natural regulatory mechanism in cells. Some metabolic pathways have built-in mechanisms for self-regulation. In them, the enzyme of the initial reaction acts as a regulator enzyme or pacemaker enzyme, and the end product of the reaction chains acts as an inhibitor of this enzyme. The regulator enzyme initiates the reaction chain, which proceeds to several intermediate reactions and products and finally yields the end product. The activity of the pacemaker enzyme in turn is regulated by an excessive concentration of the end product. If too much of the end product accumulates within the cell, the automatic control system operates to stop this further production. In this case, the end product inhibits the action of the regulator pacemaker enzyme and thereby prevents the onset of the metabolic pathway. It acts as an allosteric inhibitor which is structurally and functionally different from the actual substrate, and reversibly binds with the allosteric site or regulator site of the enzyme. This binding brings about conformational changes in the enzyme, adversely affects the reactivity of its catalytic site, and ultimately makes the enzyme inactive. This is called end product inhibition, feedback inhibition, or allosteric inhibition. So, enzyme specificity is there. Specificity is a unique feature of all living systems. Enzyme substrate specificity is an excellent example of biological specificity. Enzymes are highly specific in their action. This specificity is of two kinds, namely reaction specificity and substrate specificity. In the reaction specificity, an enzyme catalyzes only a specific kind of reaction, regardless of the nature of the substrate. But in the latter, the action of the enzyme is restricted only to a particular type of substrate. Hydrolases, dehydratases, decarboxylases, etc. are reaction specific enzymes. In the sense, they just catalyze only a kind of reaction. And whereas urease, amylase, proteases, lipases, etc. are substrate-specific enzymes. Kinds of enzyme substrate specificity. Enzymes exhibit different degree of levels of substrate specificity. 5 kinds of enzyme substrate specificity can be recognized as follows. So, first type of kinds of enzyme substrate specificity. Enzymes exhibit different degree of levels of substrate specificity. 5 kinds of enzyme substrate specificity can be recognized. Like in this case, first one is the stereochemical substrate specificity. In this case, an enzyme catalyzes the conversion of only one of the stereoisomers or optical isomers of a substrate. This is an extreme case of enzyme substrate specificity. It enables enzymes to discriminate between related substrates. The enzyme catalyzes the conversion of only one of the stereoisomers or optical isomers of a substrate. This is an extreme case of enzyme substrate specificity. It enables enzymes to discriminate between related substrates. The action of fumaric hydratase, maltase, etc. are examples. The former catalyze the conversion of only fumaric acid and never that of its isomer malic acid. The later catalyze the conversion of only alpha glycosides and not that of beta glycosides. And next is the absolute substrate specificity. In this case, an enzyme acts exclusively on a specific substrate. The action of urease is an example. This enzyme acts only on urea and it fails to act even on the slightest chemically modified form of urea. Then is the relative substrate specificity. The enzyme catalyzes the conversion of a group of chemically different substrates. For example, the enzyme cytochrome P450 takes part in the hydrolysis of a broad spectrum of substrates. This kind of enzyme constitute the least specific enzyme systems like relative substrate specificity. Relative substrate specificity. Next is the absolute group specificity. The enzyme catalyzes a particular reaction of a group or several homologous substrates. So the enzyme has absolute reaction specificity, but only relative substrate specificity. Absolute group specificity. The enzyme catalyzes a particular reaction of a group or several homologous substrates. So the enzyme has absolute reaction specificity, but only relative substrate specificity. The action of alcohol dehydrogenase is an example. This enzyme can catalyze the conversion of a variety of alcohols, though it prefers ethanol. That is absolute group specificity. Relative group specificity, bond specificity. The enzyme is reactive only to a specific bond on a group of related substrates. For example, the digestive enzyme pepsin and trypsin act on the peptide bonds between certain specific amino acids in different kinds of proteins. That is bond specificity. Absolute group specificity, relative substrate specificity, absolute substrate specificity, and the stereochemical substrate specificity. Next is the topic which is very relevant and important, that is the cofactor. Cofactor is the non-protein organic or inorganic part of certain conjugated enzymes. The protein part of the enzyme is called apoenzyme. Cofactor are indispensable for the active functioning of the enzyme. They fall under two major groups, namely organic cofactors and inorganic cofactors. Organic cofactors are of two categories, namely prosthetic group and coenzymes. Inorganic cofactors include metal ions. Prosthetic groups are the organic cofactors which remain tightly and covalently bound to the apoenzyme. The porphyrin moiety of peroxidase is an example. Coenzyme is less tightly bound to the apoenzyme and usually they can be removed by the process of dialysis. Examples are coenzyme A, COQ. Inorganic cofactors. The inorganic cofactors or metal ions that are necessary for enzyme activity are either loosely or tightly bound to the apoenzyme. Depending upon the nature of the latter, tightly bound ions are divalent or trivalent and loosely bound ones are monovalent. Some important inorganic cofactors are manganese, magnesium, molybdenum, nickel, selenium, zinc, etc. Manganese is for arginase, magnesium for polymerase, molybdenum for xanthine oxidase, nickel for urease, selenium for glutathione peroxide, and zinc for dehydrogenase. So that is something which is very relevant about the cofactors. Apoenzyme, inorganic cofactors, and prosthetic group will be there. Inorganic cofactors include metal ions.